everybody. It is my great pleasure and honor today to introduce Professor Natalia Pabón-Mora, who's going to be presenting our herbarium seminar today. Natalia is a very old friend of mine. Actually, I'm older. Let's be clear. I've known her since she was a graduate student. She was a graduate student with my best friend, Amy Licht. She did her PhD at the New York Botanical Garden through a program that they had at the time with CUNY. She did some fascinating work about the molecular and functional evolution of a gene lineage called Apodanthaceae, fruitful, which is a gene lineage that's very important for floral organ identity. And her work was really seminal in helping us to understand that that aspect of floral organ identity is in fact not conserved and is kind of like all over the place. So she really tackled the hardest question in the whole part of ABC model evolution. And when she finished her PhD, she was offered a faculty position at the University of Antioquia in Medellin, and she went back to Colombia. And when she set up her lab there, I think she realized that was this huge swath, really huge, of angiosperm biodiversity that plant Evo Devo was largely ignoring. People were working on tomato and grasses, and I'm working on temperate stuff. There's all this diversity in the tropics, and nobody was working there. Nobody really had the expertise combined with the opportunity to really work in tropical plants, and that's what she has done. And so she spent the last 12 years, 10 years, as a faculty member with a lab of undergraduates and master's students exploring all kinds of questions in tropical floral and fruit and flowering time, like all these different aspects of tropical plant development and biology. And she received, in recognition of this, the first early investigator award given by the Pan American Society for Evolutionary and Developmental Biology. And that's not just plants, that's plants and animals. So she was our inaugural winner of the early investigator award. And Chuck and I are thrilled that we get to host her this semester while she's visiting on a Fulbright fellowship, and she's going to be talking to us about this crazy endoparasite that she works on. So her talk is titled Dissecting Holoparasitic Plants, Evo Devo Case Studies in Cucurbitales and Piperales. So thank you so much.
Thank you. Thank you so much for that introduction. I am very excited to be here with you today, telling you a little bit of what we do in the Plant Evo Devo Lab at Medellin in Colombia. And I want to start by telling you, as Elena was saying, that we have tackled the genetic mechanisms underlying the formation of floral meristems, floral organ identity, carpels, and carpet to fruit transformations. Basically because these are the key synapomorphies of flowering plants, right? And they are probably the aiding source of their diversification when compared to other seed plant lineages like the gymnosperm. So they have crazily diversified in these modern environments. So over the past 10 years, because we are based on a biodiversity hotspot, we have been able to tackle a number of systems. And this is because we are trained as field botanists, but also because I literally cannot say no to new interesting projects. So it's pathological at this point. Basically, we have been interested in understanding the genetic basis of fruit and pseudofruit formation in the Chloranthacea. We have investigated the genetic basis of floral organ, floral symmetry, color, and epidermal elaboration in the family Aristolochiaceae. We are working on the genetic control of flowering time in tropical orchids. In the past, we have tackled the genetic basis of petal loss in tropical Piperales, and this is the genus Boconia. And we have studied the genes linked to bilateral symmetry and spur formation in the nasturtiums, that is the Tropulaceae family. In the Brassicales, we have studied the genetic factors controlling fruit development in a number of plant families, but we're mostly interested in Papaveraceae, Solanaceae, and Rubiaceae. And we have worked on the genomic content of these very strange, very rare holoparasitic plants, which is what I will be mostly telling you about today. And so I want to start by introducing you to the concept of holoparasitism. And it turns out that holoparasitism has occurred a number of times during angiosperm evolution. And basically here you have a first instance of that happening in the early diverging angiosperms in the order Piperales. There are no known cases of holoparasitism in the monocots, and there are 6 additional instances of holoparasitism occurring in the choriotypes. And so basically, what you're able to see from this slide is that these extraordinary organisms have lost their ancestral autotrophic capabilities, and they rely on their host for everything, right? They rely heavily on the host metabolism. And so basically that means that these plants get to tap into both the xylem and the phloem to get the water and the nutrients they need. And so for the most part of this talk, I will be telling you what we know from this family, the Apodanthaceae, in the order Cucurbitales, and a little bit of what we have learned from Hydnoraceae in the Piperales. Okay, so let me introduce you to the family Apodanthaceae. This is a family of 2 genera and 12 holoparasitic species. The monotypic Apodanthes parasitizes trees in the genus Casearia. That's a member of the Salicaceae, and it basically inhabits wet lowland forests primarily, whereas the 11 species of Phylogenetic over there are holoparasites of non-closely related Fabaceae members, so that's the legume family, and they are mostly inhabiting dry habitats. So one important feature to highlight right here is that these 2 genera have undergone extreme ecological and host shifts, and they also have different distribution patterns. So for example, Apodanthes over here, it's only found in the Americas, whereas the species of Phylogenetic occur more broadly. They are in Africa, in the Near East, in Australia, as well as in the Americas. And so basically this disjunct distribution kind of rules out long distance dispersal events as an explanation. And it basically promotes the idea or the hypothesis that this holoparasitic relationship was already in place prior to the Gondwana split. And so one other thing that I want to remark from this slide is that because we are in Colombia, we have been able to sample the full representation of this family. So here, I just want to point out the exact system that we're working with. This is a very intimate relationship between Pilostyles boyacensis, a species that we described back in 2014, with its host, Dallea quatrecasaceae. And here you can see from the photograph and from the video that I will play in the right of the slide, that this is a very intimate relationship. And here you can see that the video will start with the out of focus flower of Dallea. And basically there are 2 things that I want to highlight. The first one is that the flowering time is synchronous between the 2. So the 2 species flower simultaneously. And the other thing that I want to point out is that the infections can get so dramatic that the host eventually can die out of that particular infection, as you will see right there. Okay. And the infections are mostly at the base of the stem of that plant. All right, so the thing that called my attention the most is that Pilostyles in fact lacks all type of vegetative growth. So when you look at these plants, everything you see is the emerging bursting flowers of the parasitic plant. And so basically this means that Pilostyles does not have a root or shoot apical meristem. It does not produce any leaves. It's just basically bursting out through the bark of the host in the shape of fully formed flowers that eventually will form the fruits, as I will show you in a second. So that for us was really interesting. And I'm going to show you what's happening underneath the bark. Basically, all of these plants are growing vegetatively over here. And I want to point out that I have drawn dashes around those cells so you can see it a little bit better. The parasitic plant grows in these teardrop shaped patterns that usually accumulate around the cambial zone. They extend towards the phloem of the bark, also the parenchymatic part of the bark, but they also extend what we call sinkers or extensors, which are thin lines of cells that go into the xylem of the host. So that's important to keep in mind. And then something that might not be so obvious at this point is that all the stages of flower development in fact occur inside of the host. So that's pretty remarkable because when you think about this meristem, early meristem initiation, and all the floral organ identity are being actually acquired and formed inside of the host. And so this is, I'm showing you here the acropetal spiral sequence of perianth initiation. The outermost organs will remain purple and only the innermost will change or will show this white coloration at the end. So Pilostylis has male and female flowers as well. You can see them from the photographs right here. These are the male flowers. They don't have really typical anthers at all. They have just these pollen sacs that are forming what we call a synandrium. They actually mature from bottom to top. And it's interesting because those male flowers do have a central column, which might be a vestigial gynecium. This column has these particular vesicular hairs that produce oil concomitant to pollen maturation as well. The female flowers are a lot different. They have a short style, a massive stigma. They are formed out of the fusion of 4 carpels, but they do have a ton of little ovules that are fully formed and that have no reduction whatsoever. So they're perfectly fine. They will have the 2 integuments as expected for the group. And when these flowers are pollinated, they transform into fleshy fruits. These are tiny berries, just for you to have an idea. Everything that I'm showing you here is less than 0.5 centimeters. So all of this is really, really small. And these fleshy fruits have a ton of minute seeds that also have a fully formed embryo in their inside. So they seem to be perfectly viable as well. So pretty rare, right? On top of that, we have found what we have been calling chimeric flowers in the field. So eventually, occasionally, we do see the occurrence of these flowers. These are half male, half female flowers. And basically, you can see here that the half male produces normal pollen grains, and the half female produces regular ovules. So we don't know what that's about, but that we think is really interesting. Now, the other couple of things that I want to highlight is that pollination, seed dispersal, and germination have not been studied in depth in this system. And so basically, all we have done at this point is we have recorded the presence of these scale bugs that you see in the video that seem to be feeding off the flowers, but we don't know if these are the effective pollinators. And I hope you can see from the video at some point that there are some ants passing through. We have seen ants or ant nests associated with these plants. We do know that the seeds carry eliosomes, which are structures that have been associated with ant dispersal. However, it is also unclear if ants are in fact playing a role as dispersal agents for these particular plants. So there is a lot of field work that needs to be carried out in this system. So such a unique system kind of promotes a number of questions, right? Like we were interested in so many things, but we decided to tackle 3 main questions, which is what I will be showing you in this presentation. So the first one had to do with the genomic content. We wanted to know how the plastome and the mitochondrial genome and the nuclear genome was in this plant, if it was reduced, if we could see any evidence of horizontal gene transfer, for example. We wanted to learn more about those genes that were retained under this extreme reduction. So given the fact that we knew what are those shoot apical meristem related genes, we wanted to know if they were actually in fact retained in this plant. And finally, we wanted to learn more about the developmental gene network that was underlying flowering and floral organ identity in this unique system. And so for that, we started off by building our genetic resources, right? So the first thing we do was to generate a draft genome for this plant. In fact, this is out of the Pilostylis boyacensis floral buds that are emerging already. And we also generated a number of reference transcriptome data that basically span the full life cycle of the plant. So we have reference transcriptomes for the earliest developmental stage, that is when the endophytic growth is occurring inside of the Dahlia bark. We have a reference transcriptome for floral buds that are emerging through the bark. And we have a final reference transcriptome for those fully formed fruits that are carrying all of the seeds with the embryos inside. More recently, we have also sequenced what we have been calling, the control transcriptomes. And so those would be the Dahlia stems that have not been parasitized by Pilostylis and the inflorescence of Dahlia. And so that has been really helpful, as I will show you in a second. Now, as literally in perhaps the past semester, we have been able to collect the sister genus Apodanthaceae. That was a lot harder to collect. These are tiny little floral buds, again, less than 0.5 centimeters, but they parasitize a large tree, 8-meter tree in a low wet forest. So that was a little bit more complicated, but we generated again that reference genome and transcriptome spanning the full life cycle of this particular plant. And so I guess the first and easiest task to accomplish was to sort out the genomic compartments. And so basically here I'm showing you that based on the length and the depth of those contigs, we were able to sort out what we think it corresponds to the nuclear genome, what we think corresponds to the mitochondrial genome here in pink, and that molecule that is super highly present in the sample that corresponds to a single molecule in the plastome genome. And so here I just want to point out that we did annotate the plastome of Phylogenetic. This is a very reduced chloroplast. It's only 12,000 base pairs. It's tiny, tiny compared to non-parasitic angiosperms, which range, over 150,000 base pairs. And basically this, but it is well within the range of other Phylogenetic plastomes. And what is important to highlight is that we only were able to annotate a few genes in here, 7 genes to be more precisely, 2 ribosomal subunits, and a number of ribosomal proteins. And perhaps the only additional gene that we were able to find is this oxidoreductase, which was not found in other species of the genus. Interestingly, we did map transcription for all of these genes, which it's very interesting because it was supposed to be pseudogenized, but active transcription actually goes against that idea. So it seems that those genes are pretty active. And we know that there is high synchrony between at least 3 of the species that have been sequenced. This one is from Africa, this one is from Australia, and this is our American species. And synteny is pretty much conserved in those plastomes. The plastome of Apodanthaceae is also very interesting. It's also really reduced. It's only 13,000 base pairs. And again, compared to that in Phylogenetic, there are not very many differences. The only difference is that ACCD is complete. There is a GADPH unit and an ATPase unit. Those are the only differences. One thing that is remarkable in this case is that transcription is very low in this plastome compared to the one that I showed you. As for the mitochondrial and the nuclear genome, this is all we know so far. The mitochondrial genome is being analyzed with the help of Jeff Maurer at the University of Nebraska, and he has been able to track down a number of horizontal gene transfer instances between the host and the parasite. And this is our nuclear genome in this peak. It has been estimated in 300 megabases, which is pretty big, but not super, super big, and is well within the range of other Cucurbitales, such as Cucumis. But this genome, as you can see from this graph, is not alone. We have been able to trace also other genomes associated in this system. There is plenty of bacteria, fungi, and there is even a secondary peak of plant genome, which we think corresponds to the host plant, as this is not axenic. All right, so let me move to the more developmental part of this story, right? So we were interested in learning about those genes that are important for shoot apical meristem development. And basically, given that extreme vegetative reduction that I just told you about that occurs in Phylogenetic. And basically, I just want to kind of remind you all that the basic body plan exists because there are 2 meristems. One that is a root apical meristem that will form all the underground system, and the shoot apical meristem that is there since embryo development that will form every single aerial organ in the plant. But Phylogenetic doesn't have any of those, right? So it lacks shoot apical meristems, it lacks root apical meristems, and we were interested in investigating what was happening in that reduction, whether those genes that were known to control, say, pluripotency in the shoot apical meristem were conserved in this particular system. And so for that, we came to study those genes that have been already identified in model species. So this is Rhabdopsis thaliana. And we know in Rhabdopsis, there is a number of genes that are really important for maintaining pluripotency in the shoot apical meristem. So I just want to run through those very quickly just to give you an idea of who those genes are. And we know that, for example, WUSCHEL is a major transcription factor in the meristem that promotes pluripotency. The protein of WUSCHEL actually moves outside into the periphery of the meristem, and WUSCHEL activates its own counteracting factor, which is a small peptide called clavata3. Clavata3 activates its own receptor, which is clavata1, and basically what they are supposed to do is they are ready to repress pluripotency in order to promote lateral organ formation. That's their role. And so this, let's say, tightly regulated feedback loop is also reinforced by different factors such as BAM, that stands for barely any meristem. And on the other hand, we have WOX9, which together with WUSCHEL promotes pluripotency. So that's a whole cascade of genes, right? In parallel to this genetic network, we know that there is a gene called NOX1 that is very important for maintaining pluripotency all throughout the dome of that apical meristem. And so these genes basically belong to different gene families. Everyone is evolving in different gene lineages, but we wanted to assess whether these genes were still present and where were they be expressing in this particular system. And again, here, I just want you to concentrate on what we are calling the endophytic tissue. I'll be referring to that often. And so that is basically, again, those teardrop growth that I have labeled for you with the dashed lines over there. And so we wanted to see if these genes were present in the system, right? So our results show that in fact, WUSCHEL is present. However, kind of unexpectedly, WUSCHEL is only found in floral meristems. So it's not present in the early developmental stages that would correspond to the endophytic growth of this plant. And is not present in the fruit or in embryo development. However, and perhaps in replacement of the absence of WUSCHEL, we were able to find WOX9 in all 3 developmental stages. And even when we look at this transcription factor using in situ hybridization, we think this gene is expressed inside of the ovule in the inner integument, and we think it's also expressed in the embryo of Phylogenetic. So that's exciting, but we were not able to find any homologs for CLAVATA1 or CLAVATA3 genes. And recall that these genes are important for repressing pluripotency and forming lateral organs. So it's perhaps not that unusual that we're not finding them, but I'll come back to that in a second. We did look for them in the transcriptome and in the genome, and there's no trace of these genes in this system. And finally, we were able to recover BAM genes in all 3 developmental stages. However, as the role of BAM is to quench out the excess of CLAVATA3, it's really not clear what these genes are doing in this system. All right, so let me just give you the conclusions for this part of the talk. We know that despite that huge reduction, Phylogenetic does show at least one transcript with all functional domains for these 3 genes, BAM1, WUSCHEL, and WOX9. We know that there are no CLAVATA3 or CLAVATA1 homologs, and basically this will indicate that there are alternative repression modules for those pluripotency genes, or perhaps even better, no need for repression as there are no lateral organs being formed, right? And then finally, that we think we have found what we call a minimal genetic regulatory network. And basically, if this is true, these would stand on the shoulders of WOX9 and BAM1, as these are the only 2 genes present in that early developmental stage. However, I do want to point out that we know that there are NOX genes in this system and that we're only starting to address their evolution and their expression patterns. But we know the NOX cascade is also present in Phylogenetic. Okay. So what about the genes that are important for flowering and floral organ identity? And this was kind of my jam at the beginning. That's what I really wanted to get at to start with. And so here I just want to remind everyone that, again, despite that huge reduction in the vegetative growth, these plants have perfectly normal floral meristems with all floral organs formed, and they produce normal flowers and normal seeds. And so that part of the life cycle is, say, intact. Okay, so flowering is a very complicated process in terms of the molecular underlying genetic mechanisms. And I just want to point out a few general aspects. So the first one is that flowering requires a lot of signals in place. It requires a particular age, a hormonal stage, and it relies on how much light or the temperature that the plant is receiving, which we call the photoperiod and the vernalization. And this process starts out with this molecule FT, that is synthesized in the leaves, but moves through the phloem towards the shoot apical meristem, and there it triggers what we call the phase transition, right? The reproductive transition of the plant. And FT does so by activating this cascade that I have placed over here, including AGL24, SOC1, and LEAFY. And LEAFY is in charge of generating the identity of those floral meristems. So those are what we call the flowering integrators. Those are the positive integrators. And if anything happens to them, the plant will basically not reach the flowering transition. On the other hand, there are the negative regulators. They include SBP, TFL1, and FLC. And if anything happens to those, as they negatively regulate that process, then those plants will actually in fact flower faster or earlier. And I just want to remind everyone that these genes are part of different gene lineages again. But something very interesting happens in this system as closely related genes that are part of one single family can be doing completely opposite function during flowering transition. So for example, say TFL1 and FT in the PEBP family, they do opposite functions during this process. And the same happens with these matchbox genes over here. Okay, so I want to show you that we reconstructed the evolution of every single one of these lineages that are part of the flowering integrators. And I just want to point your attention to the stars. So basically, every time you see a star, there is an instance of a contig that was recovered from the Pilostyles transcriptome that nests within Fabaceae homologs. Okay, so this is one example of that. I hope you can see that. So that's the gene from Pilostyles and is nesting with Medicago, Phaseolus, and soybean, right? So basically, this would point to the idea that the parasite is recruiting the host signal in order to flower, right? And so that is interesting. That is actually not new. It has been reported at least in this plant, which is Cuscuta. This is what is broadly known as the daughters. And those plants actually can invade or can parasitize different systems and adjust their flowering time to the one that their host has. And so that has been shown to occur at least for FT in this particular system. But in this case, a scenario, we not only have FT doing so, but all of the other flowering integrators, perhaps with the exception of one single copy of SOC1. So that is pretty remarkable. Those experiments are really elegant, but we are unable to move forward with any type of experiment like that because this holoparasitic plant only parasitizes Dalea or Fabaceae species, right? And in this case, only Dalea quadricasaceae. So we thought one way to address this was to investigate the expression of FT in both the host and the parasitic tissue. And so far you have seen kind of the cute, the tender, soft side of the host, but don't be fooled. These plants are made to survive the drought. And so basically these plants have a ton of sclerenchymatic tissue in the bracts and in the sepals. And on top of that, they produce these oil glands that you can see with the yellow arrows. And so this is not precisely the type of plant that you would want to have for molecular work, right? But patience comes a long way. And I'm going to show you that we have been able to find the expression of what we think is the FT of the host, okay? So this is expression of FT in Dalea quadricasaceae is very low in the inflorescence meristem, but it does seem to be turned on in the lateral floral meristems, which is pretty typical for FT and also pretty typical for other legumes that have been investigated. The level of FT drops down as the flowers mature, and that's what we see also in this system. So wouldn't that be interesting if we could find FT of the host in the parasitic plant? That would be great. And that's exactly what we have been finding so far. So this is the expression of FT of the host, but this is the parasitic tissue. And you can see that the signal is, it's still, we're still working on the controls. You can see the signal, but it's very much in the line of the growth of the Pilostyles parasitic tissue. And so you see that the signal comes on very early and it starts to drop down. It's only really maintained in the vascular tissue at mid-developmental stages. Eventually, it's sort of in the vasculature, but it does drop down after the floral organs are fully formed in the parasite. So that is really interesting. It would point out to the fact that the mRNA of the host is somehow being moved or shared between the two plants. That is exciting. That will require a lot of biochemical controls that we so far don't have. But by itself, it does not explain how flowering occurs. So I want to point out to you that in all species so far investigated is actually the relationship between FT and FD, what triggers flowering. And so far, we have not been able to find FD in the parasitic plant. So in Cuscuta, FD is present in Cuscuta, and the FT of the host moves, and it interacts with the FD of the daughters. In this case, we have been unable to find the FD homolog. Now, I want to point out from this diagram that basically everything that you see, this is the flowering integrators all together, and everything that you see in green corresponds to a gene that was taken or found in the Pilostyles transcriptome, but in fact belongs to the host plant. So there is a heavy reliance of the parasite to the host in order to flower, right? Pretty much everything comes from the host, except for SOC1 and LEAFY. And this is only one copy out of all the SOC1s. Okay, so what about endogenous factors? Can we find anything that the parasite is actually doing by itself? And the question is, for sure, there is LEAFY. And LEAFY is actually one of the most important markers, as I mentioned, for floral meristem identity. And the only thing is that LEAFY is expressed very early on. Even in the young endophytic tissue of this parasite, you can see the expression of LEAFY in those teardrop kind of early growths. You can see it here in a little bit more detail. That's at 40x magnification, but you can see how LEAFY is expressed very early on as it bulges out into the phloem and the parenchyma. And it's even expressed there as the floral meristem grows, right? So LEAFY is definitely endogenous, is definitely being produced by the parasite, and is broadly expressed at least into this stage. Eventually, again, it drops down in its expression in the outermost floral organs, but it's retained in the innermost floral organs, such as the stamens and the carpels. Okay, what about the genes important for floral organ identity? And so here I just want to point out that all of those genes have been previously identified in Rhabdopsis thaliana. I'm sure you have heard about these genes at some point. These are important for the establishment of the identity of each organ. And basically the model kind of consists of this interaction between these four classes of transcription factors that come together in order to provide the identity to a specific floral organ. And so as the model would predict, A and E genes together control sepal identity, A, B, and E genes together control petal identity, B, C, and E class genes control stamen identity, and C and E class genes control carpel identity. And basically what we did was to sample all of these genes in the system, and there is very little surprise here. All of the genes that you see with a check mark have been isolated from the parasitic plant, are endogenously produced, and they seem to complete the full set of matchbox genes that are important for floral organ identity. So it seems that after LEAFY is turned on, everything is done autonomously by the parasite. All right, so we just want to give you the conclusions for this part. The floral promoters so far identified, including FT, AGL24, and SOC1, likely belong to the host, but are being used by the parasitic plant. Same happens with the only flowering repressor, that's SBP, is the same story. It belongs to the host, but it might be used by the parasite. LEAFY is endogenous in Pilostyles, and there is something really interesting here. It kind of supports that early expression that I showed you. It kind of supports the idea that has been put forward by other researchers working in other systems like Rafflesiaceae. So, for example, that idea that heterochrony is linked to fast flowering in endoholoparasites would be supported with this early expression of LEAFY in that tissue. And Pilostyles has a complete set of floral organ identity matchbox genes, and that includes the B class genes, AP3 and pistillata, the C class gene agamus, the E class genes, 2 of them sepallatas, and 1 C. So that's pretty much set. All right, so for the rest of this talk, I want to kind of move a little bit just to show you what we have learned of a different system in the Piperales. And so the Piperales is an order that we have worked on for a long, long time. It comprises 4,000 species, and it can be broadly divided into 2 groups, those without perianth and those with perianth over here. And something that called our attention, again, very early on when we started the lab, was that particular difference in terms of perianth morphology that the genus Aristolochia has when compared to other taxa in the order, okay? So we started working in Aristolochia, and it turns out that Aristolochia is also the most diverse genus. It comprises around 550 species, and you can just see how the perianth morphology is so incredible and so diverse. And we were really interested in tackling these questions about like where did that perianth was coming from? How is it being transformed? If there were different colors, why would that be? And how was that acquired during flower development? And so basically we started over here with these questions. We knew from early observations that Aristolochia plants do not have true petals. Their perianth is formed out of 3 sepals that fuse together very early on. And in these plants, like I said, there are no true petals, but there is a transfer of function as the sepals kind of become petaloid as they go along. And so you see that they accumulate colors, pigments. They can be in the ranges of yellow or sometimes purple or sometimes both. And they also have populated cells and trichomes in the perianth. And not only that, but Aristolochia flowers form this unique structure formed out of the congenital fusion of stamens and the stigmatic tips of the carpels, and that is called the gymnostigma. So there was plenty of questions that we wanted to address again, but we concentrated first to generate genetic data that we could use. We generated reference transcriptomes not only for this fast flowering species, which is Aristolochia fimbriata, which you might have seen, but also for close relatives in the genus Aristolochia, but also in different genera, including Saruma, Sarum, Totea, and others. And so this was done with the help of the Technical University of Dresden and the Arnold Arboretum here in Harvard. And so basically our pipeline kind of consisted on searching homologs that were interesting for a particular developmental question. Then we used to do phylogenetic analysis to assess their homology. And then we proceeded to study them with expression analysis, whether that was using RT-PCR or qRT-PCR or in situ hybridization. And more recently, we have integrated into these studies RNA-seq data. And so when we started the lab, that was our first question. We started to tackle the same genes, the ABCE genes in the model for floral organ identity in Aristolochia. And I will just show you briefly what we found. We knew that Aristolochia fimbriata had copies of or homologs of all of these genes, a single copy of everything except sepalata, which is not a surprise. Sepalata genes have duplicated very early on during flowering, during angiosperm diversification. And we generated this data set exploring the expression of these genes. And so basically, I'll just run through it really quickly. I'll tell you that agamos is expressed in stamens and carpels. That's pretty much expected, as the canonical role is in Arabidopsis. We think these genes are also performing stamen and carpel identity. Seed stick is a D-class gene, is expressed in the stamens and in the ovules, also again, pretty much expected. The only weird or new thing is that seed stick in this case is expressed in the dehiscence zone of the fruit. The B-class genes had been studied previously to these analyses. These B-class genes are expressed in stamens, as other studies had shown. And this basically corroborates the idea that the perianth is sepal-derived and not petal-derived in this taxa. And we knew that the expression patterns of sepalata were very low, but very broad. Something similar with full broad expression patterns, again, even in the shoot apical meristem and in the leaves, suggesting more pleiotropic roles for this gene, and not so much a floral organ identity function. But perhaps the most interesting result that we got, that was back in 2015, was the expression of AGL6. AGL6 had not been placed or associated with the floral organ identity at that point. And we knew that AGL6 was important in perianth development. It was a gene that was turned on very early on and was retained during perianth development throughout flower development. AGL6 is also expressed in ovules, which is where it's expressed in Arabidopsis, suggesting that this gene was already performing a number of functions early on in flowering plants. And so we went a step further. We generated the data to test protein-protein interaction, as these genes are known to perform their function in tetrameric complexes, meaning they have to touch each other in order to perform that role. And we generated this data where we thought perhaps AGL6 and Sepalata2, that dimer or that heterodimer was the one responsible for parent identity. And at that time, we contributed to filling this gap that was missing. There was a lot of data in choriodicots, in monocots, and even in aquilegia, but really nothing in the early diverging angiosperms. And so that's where we started. We, since then, have tackled a number of questions. So we have explored the genetic basis for bilateral symmetry in these plants. We have explored the genes that might be important for epidermal elaboration, meaning the formation of trichomes in these flowers. We have started to explore some of the genes that are important for floral coloration in these plants, and also which genes might be important or modified in this crazy gynostemium that these plants have. But nothing prepared us for this monster, okay? So basically, this is Hydnora, right? This is the only holoparasitic member of this order. And what I want to just kind of highlight is that this project came to us by through our colleague, Stefan Wanke. He is a professor at the Technical University of Dresden. This is Stefan collecting with his friend James Bowling in the deserts of Namibia. And basically what you're seeing here is Hydnora vicerii, and that is a holoparasite or a parasite of this shrub in the back, which is a member of Euphorbia. And so what he's holding in his hands is the vegetative growth, that rhizome that is kind of growing very close to the soil in this system. And so I just want to kind of point things to you because it wasn't easy at the beginning for me either. This is the rhizome that grows close to the soil. That rhizome will eventually branch out and produce these floral buds that are emerging. And those floral buds will open and pollination will occur with the help of beetles and be aided by thermogenesis produced in the inside of the flower. And then eventually these floral buds will form those fleshy fruits that are about 15 centimeters. So that is insane, right? Okay. So what Stefan did was he collected, basically he went to the field and he collected the tips of the rhizome, floral buds, he collected the tepals. In this case, he collected both the fleshy portion and the heating part of the tepal, I guess that's called the osmophore, stamens, carpels, and fruits. And he passed on that data to us. And basically what he wanted to know is whether Hydnora had or didn't have the ABC DE genes of the floral model, right? But we thought this plant was so unique and so bizarre that we just extended the sampling and we generated a whole data set, not only for Hydnora, but all other members that we had for all of the MIKC star genes, Matchbox genes. And so basically, I'll just run through it again just to show you what we found. Everything that has an arrow in there was found in Hydnora. So basically, Hydnora has a full set of B-class genes. It has agamos, which is the C-class gene. It has seed stick over here. It has a full homologue. It has Sepalata genes. It has AGL6, and it has a SOC1 homologue. I want to draw your attention to the fact that it does not have a SEP3 ortholog. So that's interesting because that was supposed to be like the most important gene for floral organ identity, but I guess Hydnora has a backup plan or something with the other copy right there. But that was the only difference we found. All of the other genes that don't have the arrow we weren't able to find any homologs in the system. In terms of expression patterns, there is really not, or there's very little novelty here. So for example, Hydnora also is using the same candidates as I just showed you in Aristolochia for parent identity. It seems that AGL6 is also pretty active in this plant. And again, for a stamen and carpel identity, Hydnora is using the typical B, C, and D class genes. So that is very much as the expected pattern. One thing that I want to show you is that we compared again all of the flowering integrators from Hydnora to those of temperate and tropical Aristolochia species. And I just want to tell you that everything you see in green is produced autonomously, meaning that Hydnora is able to produce a lot of the flowering integrators by itself. which is very different from what I just showed you in the other system. Okay, so this is less dependent, I guess, from its host. The only things that Hydnoraceae is missing are phytochromes, which actually blast with host genes, and I guess a number of genes important for the circadian clock and the autonomous pathway. But everything down the line that integrates flowering is there. I also want to point out that we have not been able to find FB in this system. Okay, and finally, just to kind of close up with this data set, I wanted to tell you that we did find some evolutionary traits with huge implications for parent development in this study that we did, just because every single gene lineage that has been associated with parent identity so far, including AGL6, SEP2, and FUL, is duplicated in every species that has a B part-type parent. So this is something that we did not know. Both Asarum and Saruma have 2 copies of each of these genes, whereas the unipart-type parent species, including Aristolochia, Hydnora, and Totea, only have a single copy. So this is worth investigating further down the line. All right, so I just want to drop a few conclusions. Here, Hydnora is definitely more genetically independent from its host. And this is likely, I would say, due to the presence of this rhizome, right? So there is a whole vegetative body there, and perhaps it even has phyllotaxis, who knows? So that might explain this difference. The flowering integrators are in fact endogenous in Hydnora. However, the host might be providing some signals, perhaps phytochromes, perhaps some of the autonomous signals. And I just want to point out that floral organ identity in Hydnora relies on the exact same transcription factors that were already identified in Aristolochia, perhaps with the exception of SEP3. And kind of a closing remark, I guess, for this presentation is that something to conclude from both portions of the talk is that I guess the more structural reduction occurs in the endophyte of different holoparasitic plants, the less genetic autonomy there is for reproductive transitions, right? So I guess that would be my take-home message in case you want to tell others what you learned today. All right, and so with that, I just want to acknowledge everyone for being here. I want to acknowledge my hosts through this Fulbright Scholar Program, Elena Kramer and Chuck Davis. They have been very supportive with this very strange project that I brought to them. I want to thank the Kramer Lab members for all their company and their help and all the fun in the lab. And I want to thank all of our funding sources, our national collaborators. This is Fabio Gonzalez, Juan Fernando Alzate, and Marco Giraldo. They have been really important for the development of every single thing that I showed you today. And this is our really large crew of international collaborators that have been very supportive since day one, since I started back in Colombia. Thanks to them, we have been able to survive all this time as a lab. And I want to finally thank my group. This is a very large group of, like Elena said, undergraduates and master's students primarily, but they are the source of continuous joy and productivity in the lab. So I want to thank them, and I want to thank you for your attention, and I will be happy to take any questions that you may have, or if you're too shy to make the questions here, you can also write me to my email. Thank you. Molly, thanks for that very important question. We don't know. So we know that there is no lateral connection between those teardrops, right? So maybe there is very little connection, but they do form, I don't know if you saw like lines of flowers. So when they emerge, they have like these lines, right? So the connection might be going that way, through the vein or through something that is indicating a particular line. We don't know how the infection occurs in the field. It might be something that starts very early on in the plant. So maybe a single infection results in all of those individuals of Piperales growing inside of a single stem. That is perhaps the most likely scenario. So in that case, everything that one sees in the layer stem would be a clone. But at this point, we don't.
I have a maybe question. So how do they disperse if they're so host specific? They have to find another legume.
Right. Right. So that's interesting. So oh yeah, sorry. So the question was, how do they disperse out? How do they find a host? And so basically, when you see the Dallea populations, they're pretty broad, they're pretty big. So once there is one infected, it's very likely that the others will eventually get it just because there's so many of them, right? So the host populations are pretty big. However, we don't know what moves the plants from the parasitic plants from one plant to another. The most likely scenario is that the seeds are in fact being dispersed by the ants. There is definitely something going on with those ants because the ants are very abundant in these populations of plants. So basically every time we see an infected Dallea plant, there is a nest close by, but no one has recorded this. So don't say anyone that I told you that. It might be that that's the most likely scenario, but we don't know what disperses out the seeds.
Has put on convergent and atypical direction, and we mentioned about Hydnoraceae, right? Now, those which have vegetative stems are underground, then compared to parasitic lacking in stems. So, do you think which is ancient or which is
That's a great question.
And then the holoparasite with the cell machinery of the host plant. But you mentioned about, using the host genome.
Right.
And just stretching the bacteria page, manipulating the bacterial cell machinery by multiplication. Here it is something using for flowering mechanism. So, these are the two questions.
Right. Okay, so the two questions are, the first one has to do with the reduction in casita and cuscata. Yeah, so I mean, right. So they have something that you can see as something that grows as a vegetative phase. So there's definitely large differences between Pilostyles and those systems. And I think that perhaps this large reduction, I haven't found anything as reduced as Pilostyles, perhaps with the exception of Rafflesia. So the endophytic tissue of Rafflesia looks very much like the one of Pilostyles, right? So they also have this teardrop, although skinnier, I guess, cell lines, which put sinkers into the xylem and grow very much like the one that I just showed you, perhaps just a little bit tighter. And I would say, since holoparasitism has evolved so many times, then these two are the most reduced, but they have converged into that. So they have nothing in common, I guess. They just converged into that shape. And that is very different from hemiparasites, for instance, right? Because they still have a lot of the vegetative body in place. Now, as for the recruitment of the host genes, so, I mean, this is all very new to me as well. So I guess this is, we're only starting to scratch the surface of how is it that these plants actually use the host genes. The interaction is very tight. So we have seen, for example, that those cells can invade cells from the host. And so there is an intimate contact there. And so it might just be that, if those things move through plasmodesmata, then that would be it, right? But there is a lot more to be learned at this point. I don't think we have enough evidence to say how is it that they're recruiting these genes. Yeah.
So I think we touched on this before, but now I can't remember. So with the Pilostyles and all of these genes from the host, what percentage of them appear to actually, or do you have a sense of what percentage of them are actually transferred into the parasitic genome versus kind of just like, oh yeah, you've got some tea in that bowl, I'm not gonna take that, response.
Right. So the question is to what extent do we see that the host genes are in fact transferred to the Piperales genome, or they're just being used or captured in the system? And so far, I have only looked at 4 of them in the genome. So a number of those flowering integrators, including FT, none of them is in the genome. So the genome is 400x deep. So we have a ton of data.
Yeah.
And none of them is in the genome. So I would think that this is more of a sequestering, I guess, the mRNA. I was actually looking into this the other day, and there is a whole paper on mRNA moving through the phloem that has been characterized in Arabidopsis that was not known to be mobile. So maybe these signals actually can move through the phloem. I just don't know enough about each of them to be sure that they can actually move.
And my other question was about the Hydnora, the rhizome. What are all the little bumps?
Oh, so I recently learned that this rhizome kind of grows kind of in the periphery of the plants, right? But then as it grows, it forms those serial buds that are supposed to be dormant meristematic tissue. And it's unclear to me when those buds become flowers. So they have been described. Exactly. Right. So that's why I even suggested that it does kind of seem to have phyllotaxy, right? Like there is this dormant buds kind of waiting to become a flower, but not all of them become flowers. So the rhizome, it actually branches quite a lot. So when they kind of untangle those plants in the field, that's a great question. It would seem from the pictures that it is, but I have never seen the plants myself.
And is that also producing a tissue that is entirely internal to the host? So it has like being internal, or is it just kind of like mosquito where it's like sticking it?
No, so I think there is an endophytic tissue, which has not been sampled then. But it might not be as reduced. It might be more like, oh, let's see where the euphorbia plants are to kind of enter and take the nutrients out.
So it might be a little bit more like
Yes, in a way, but it has been described as a holoparasite. So perhaps not a hemi, but a more divergent holoparasite.
And it's like you control like for a healthy plant, like if the moving is a healthy plant, or is the parasite like actually altering like the patterns of transportation of molecules?
Right. Okay, so that's a great question. So is FT moving in the host? So we think it is, as in every other flowering plant. So FT is a mobile signal that has been characterized as a mobile signal everywhere people have looked. We actually haven't tested that, right? Like we think it's there, we know it's flowering, we know it's in the right place, and we are assuming that FT moves in the host. And therefore, its movement through the phloem might be the one that is capturing or sequestering the parasitic plant to flower itself. Yeah.
I think the host plant is not the host plant.
The host plant is a euphorbia. Yeah, there are 12 species of Hydnora, and I don't think everything is Euphorbiaceae. There's 2 more families that, yes, I think so, yeah. I have never seen them in the field myself, but that's how it has been described. There are 2 more families that can host Hydnora. I'm just forgetting at this point. So it's not only Euphorbiaceae, there are 2 more.
Things that are necessarily transferred or kind of borrowed or used. And what is flowering? You're being described as the other also flowering.
Right. So they have synchronous flowering. Yes. Yes.
And then never the beans that are next to the host plant, are those relatively similar in size or is there?
That is a great question. We have only done maximum likelihood analysis so far, and they do seem to have large branch lengths. But I'm not sure about the exact timing or how to measure like mutation rates that would indicate how far apart they are. I mean, if it's from the host, it should be just basically from the layer, right? So it's not like a super large branch either. It's just a little bit different from the other legumes. So I would think that there is not that much difference, but yeah. Thank you.